CRISPR technology acts as molecular scissors, enabling precise DNA editing. It targets specific genes, minimizing unintended changes. Beyond human DNA, CRISPR's applications span agriculture, conservation, and disease management. Base editing, used in KJ's treatment, changes a single DNA letter without cutting, reducing risks. This precision marks a significant advancement in genetic therapies. CRISPR technology functions as molecular scissors, enabling precise DNA editing by cutting at specific sites to modify genetic sequences. Its targeted approach minimizes unintended changes, unlike earlier gene therapies. Beyond human DNA, CRISPR's versatility extends to agriculture and disease management. Base editing, used in KJ's case, alters a single DNA letter without cutting, reducing associated risks. KJ Moldun was diagnosed with CPS1 deficiency, impairing nitrogen processing. Researchers swiftly developed a CRISPR-based base editing treatment, targeting his specific mutation. In May 2025, they corrected a single DNA error, enabling KJ's body to process nitrogen effectively, curing his condition and allowing normal development. Scientists sequenced KJ's genome to pinpoint the single DNA error causing his CPS1 deficiency. They developed a base editor, a precise tool that alters one DNA letter without cutting the helix. Delivered via a modified virus targeting liver cells, the editor corrected the mutation, enabling proper nitrogen processing. The journey from diagnosis to treatment for CPS1 deficiency was remarkably swift. Within just 6 months, a custom base editor was designed, tested, and approved. This rapid development, from genetic testing to treatment administration, marks a significant advancement in personalized medicine, drastically reducing timelines that traditionally spanned years. CRISPR-based therapies offer hope for over 400 million people affected by rare genetic disorders. With 95% lacking food and drug administration-approved treatments, conditions like cystic fibrosis and sickle cell anemia could benefit from personalized genetic editing. KJ's treatment exemplifies the potential to address thousands of single-gene disorders, transforming lives globally. KJ Moldun's treatment echoes humanity's quest for healing, transforming ancient myths into reality. From divine interventions to alchemical pursuits, history is rich with tales of miraculous cures. Today, CRISPR technology marks a genetic revolution, allowing us to edit life's fundamental code, bridging the gap between myth and scientific medicine. CRISPR's potential to eradicate genetic diseases and offer personalized treatments is revolutionary. Yet, concerns about unknown long-term effects, unintended changes, and the risk of designer babies persist. The ethical debate mirrors past controversies, highlighting the tension between innovation and moral responsibility, as seen with in vitro fertilization's evolution from controversy to acceptance. Religious perspectives raise concerns about humans assuming divine creative powers through genome editing. Ethicists question the distinction between treating diseases and enhancing capabilities, pondering where society should draw the line. Natural order arguments highlight the evolutionary complexity of the human genome, cautioning against unforeseen consequences of genetic alterations, while acknowledging medicine's history of intervening in natural processes. CRISPR's precision is not absolute. Off-target effects can occur, leading to unintended DNA edits. Base editing reduces this risk, yet scientists remain vigilant. Cellular mosaicism presents another challenge, with mixed cell populations potentially impacting treatment outcomes. Additionally, genetic modifications may have delayed or intergenerational effects, necessitating long-term monitoring. Complex gene interactions further complicate predictions, highlighting the need for cautious advancement. The cost of developing genetic treatments like KJ's, exceeding $2 million, poses significant financial barriers. Advanced technologies are often concentrated in wealthy nations, exacerbating geographic disparities. Regulatory hurdles and the need for specialized healthcare infrastructure further complicate access. These factors risk widening health disparities, potentially limiting life-saving genetic cures to the affluent. Long-term monitoring is crucial for understanding the full impact of genetic treatments. Immediate effects require monitoring for efficacy and complications. Developmental impacts involve tracking growth and milestones. Genetic stability demands regular genomic analysis to detect unexpected mutations. Reproductive implications focus on potential effects on future generations. This case initiates a longitudinal study to uncover these unknowns. Historically, groundbreaking medical technologies like vaccination, anesthesia, in vitro fertilization, and organ transplants faced initial fears, ranging from social degradation to spiritual contamination. Yet, they revolutionized healthcare, eradicating diseases and extending lives. CRISPR, despite current controversies, might similarly transition from skepticism to a standard, transformative treatment. The mad scientist trope, popularized by stories like Frankenstein, contrasts sharply with reality. Modern genetic research, including CRISPR, is highly regulated, involving collaborative teams and strict oversight. Ethical committees and regulatory bodies ensure safety and transparency, emphasizing caution over recklessness. This structured approach counters the myth of isolated, unchecked scientific endeavors. The global regulatory landscape for genetic research is complex. In the United States, the Food and Drug Administration oversees somatic gene therapy, prohibiting germline editing. The European Union enforces strict regulations, banning heritable genome modifications. China's framework is evolving, with past controversies in germline editing. Internationally, the World Health Organization calls for a global governance framework to ensure ethical research practices. The debate on designer babies intensifies as CRISPR technology advances. While treating fatal genetic disorders is widely accepted, the potential to enhance physical traits or intelligence raises ethical concerns. Critics warn that these capabilities could shift focus from medical necessity to non-medical enhancements, blurring lines between therapy and enhancement and challenging societal norms. CRISPR's potential extends beyond human health, revolutionizing agriculture by enhancing crop nutrition, yield, and resilience against pests and climate change, crucial for food security. In disease vector control, gene drives could eradicate malaria by altering mosquito populations. Conservation efforts benefit from genetic rescue and biodiversity enhancement, though ethical concerns about ecosystem impacts persist. The scientific community is divided on CRISPR's pace and scope. Some celebrate KJ's treatment as a milestone, while others urge caution. Dr. Aisha Rahman highlights the complexity of editing the human genome without a comprehensive guide. Dr. James Chin argues for balancing ethical concerns with alleviating patients suffering from genetic diseases. Balancing progress and caution in CRISPR involves rigorous research, ethical frameworks, and global governance. Interdisciplinary studies enhance understanding, while evolving ethical guidelines ensure diverse perspectives. International oversight prevents misuse, ensuring equitable access. Public engagement through education and transparent communication is crucial. KJ Moldun's case highlights the promise and responsibility of this transformative technology, demanding thoughtful stewardship. CRISPR gene editing is revolutionizing medicine by offering new hope for genetic diseases. However, it presents ethical challenges, demanding careful evaluation of its societal impact. Inclusive decision-making is essential, involving diverse stakeholders to guide its development responsibly. KJ Moldun's story highlights the ongoing journey in our biological evolution.